Hello everyone. Last lecture we talked about parts that makes the immune system, the organs, the cells that are involved in making the immune system. In this lecture we are going to start talking about the pathways that the immune system follows or uses to conduct its function. So our lecture today is about innate immunity. So we have two forms of or two pathways that the immune system follows to conduct its function. The first pathway is the innate immunity, and the second is adaptive immunity. The innate immunity is sort of primitive immune response, so it does not have that complexity. It does not have the ability to recognize different types of antigens, so it has no specificity. And it does not have the ability to memorize and recognize the antigens that it has encountered before. So each time it encounters this antigen, it will follow exactly the same way in fighting that antigen. So there is no memory in this type of immunity. And as it is so primitive, it is present since birth. There is no need for learned behaviors or no need for any modulation to conduct this immune system or immune response. It has three components. It has the physicochemical, the humoral, and the cellular. In this lecture, we are going to talk about the first two components, the physicochemical and the humoral. And in the third part, we will talk about it in details in the next lecture. Starting with the physicochemical part, so the innate immunity forms the physical barrier against the entrance of the pathogen inside the body, and this involves the skin, the mucosa lining, the saliva as it washes the bacteria and antigens and debris from the tooth surfaces and from the oral mucosa, the tears that wash out and wash away all debris and dust from the eyes. So this sort of physical barrier, and we can appreciate the importance of this barrier as increased chance of developing an infection when we have a cut wound, or for example in the case of burn where a huge amount of the skin is lost. So in this case the body would be at high risk of developing infections. The chemical part, it involves the presence of chemical substances like enzymes or proteins or immunoglobulins that attack bacteria. This is present in the saliva, present in the tears, and also the pH, the low pH environment that is present in the stomach is the very important line of defense that attack all the bacteria that comes with the ingested food, and most of them will be destroyed in the low pH environment in the stomach. The presence of the normal flora, this is sort of a collaborative relationship between the human body and the certain types of harmless bacteria. So the bacteria or the body would provide the habitat for this bacteria, would make orders for the immune system to ignore the presence of these bacteria and recognize them as sort of self. So they would not be attacked, they would not be involved. In return, this bacteria would prevent the attachment of any more pathogenic bacteria to the surfaces of the gut, for example, or the surfaces of the oral mucosa. So this is the first component of the innate immunity. Moving to the second part, which makes most of this lecture today, is the humoral part of the innate immune response, and this involves type 1 interferons, microcidal molecules, complement system, opsonins, cytokines, and chemokines. We're going to talk in more details about these components of the humoral innate immunity. Starting with type 1 interferons. So we have two types of interferons. We have type 1, that includes alpha and beta interferons, and this is part of the innate immunity. And we have type 2 interferons, which is interferon gamma. Interferon gamma is part of the adaptive immune response. Talking about the type 1 interferons, alpha and beta, they are structurally exactly the same, and they conduct exactly the same function, but they are produced from different types of cells. Interferon alpha is produced by different types of white blood cells, mainly from dendritic cells, while interferon beta is produced mainly from fibroblasts. How does it work? Interferons, as I said, particularly interferon alpha, is produced from different types of cells. This image shows us the epithelial cells that could get attacked by viruses, and once the virus gets inside the cell, this would stimulate the cell to produce interferon alpha. If this happens to fibroblast cells, the fibroblast cells will start producing interferon beta. All cells in the body, they have interferon receptors. So whenever there is a viral attack in any cell, and this cell starts producing interferon alpha or interferon beta in case of fibroblasts, these interferons will bind with interferon receptors that are present in adjacent cells. This will act as an alarm that there is sort of a viral attack somewhere next door. So these adjacent cells will get prepared by producing antiviral proteins. If a virus is spread to the adjacent cells, now these cells are ready to face that virus because of the antiviral protein that will start destructing that virus, preventing it from getting into the adjacent cells. So interferons act by preventing the spread of the viral infection. Of course, the cell that was attacked first by the virus and that starts producing interferons these interferons will also attract natural killer cells. It will act as an attractant to the natural killer cells, giving them the message that this cell is infected with a virus and it needs to be destroyed. So the virally infected cells will be destroyed by natural killer cells. Other cells will be protected because they've got the message when the interferon alpha or beta bound to the receptors on the surface, and they have produced the antiviral protein that provided them with protection against viruses. This is the first mechanism the interferons work. The other part is that the interferons would also stimulate other white blood cells like macrophages, dendritic cells. They would start informing them that they need to recruit other cells if needed to control this viral infection. So this is the first part of the humoral immune response, the interferons. The second part is the microcidal molecule, and the best example of microcidal molecule is a defensin. Defensin has a unique structure. It has the lipophilic part of it, and it has another part which is charged. The lipophilic part of the defensin, it will allow it to get through the cell membrane, which consists of a lipid bilayer, so it can get inside the cell membrane, and because of the charged part here, they are positively charged, so they would move away from each other because of the repulsion forces. This would create a hole in the cell membrane. So this is a cell membrane of a pathogen. It was recognized by the defensin, the defensin is attracted to the lipid bilayer. It will open a hole here. This hole will allow the fluids to come inside, and this will lead to the burst of the pathogen. So this is another mechanism of the humoral immunity, which is the microcidal molecules. As you can see here, the defensin did not recognize specific type of antigen. It did not conduct its function based on a certain epitopes or certain parts of of that specific antigens. It was just attracted to the lipid bilayer, which is present in most of the pathogens. The third part is the complement system. So complement system is sort of more complicated than the previous two forms of the humoral immune response we talked about. Complement system, the name, as it implies, it is called complement because it complements the action of the immune system. So it is immune system still can function without the complement system, whether cellular or humoral immune system, whether innate or adaptive, still the immune system can function. But the presence of a complement system or complement proteins, this will make it more effective in conducting the function. This experiment shows the importance of the complement system. So if we injected a bacteria into a mouse and left that for a couple of weeks, now the immune system in this mouse body will form antibodies against specific and against these type of bacteria. If we remove the serum would contain the antibodies and would contain other components, other proteins. If we got this serum and we add it to another test tube that includes the bacteria, the antibodies would bind with the antigens in the bacteria, and these proteins will enhance the lysis and damage of these bacteria. They made an experiment. They took exactly the same serum, but they heated it for certain period of time. This heating did not affect the immunoglobulins, so still have the immunoglobulins intact, but the other proteins here were destructed. So we have now in this test tube, we have only the antibodies specific to that antigen, and they mix it again. These antibodies, they mix it with the antigen here. So still the antibodies could bind with the antigens, and this will lead to agglutination or aggregation of the bacteria, but no lysis took place. So these small proteins are complement proteins, and here it shows the importance of the complement proteins in finalizing and completing and complementing the function of the immune system. Here the antibodies were still there, but the job was not completed, was not finished. So these proteins are complement protein. There are more than 40 types of proteins produced by the liver, and they move through the circulation to the different types or sites of infection. Of course, when they are produced from the liver, they are produced in a non-active form. Otherwise, it will initiate an immune response wherever it goes. So they should be present. They should be in non-active form once produced by the liver, and they will be activated only when there is a pathogen, when there is a trigger for an immune response. Activation took place in different pathways. There are three pathways. The first pathway that was discovered is called the classical pathway, and later on, when they discovered that the complement system can be initiated in an alternative way, other than the way they discovered initially, so they called this the alternative pathway. Later on, they discovered another way, or another pathway, where the complements can be activated. And in the third pathway that was discovered, a mannan-binding lectin protein was involved. So they called that a mannan-binding lectin pathway. This image shows us the two different pathways that is used to activate the complement protein, the classical pathway and the alternative pathway. We don't need to go into much details and in understanding the complement system cascades. However, I'm going to give you some sort of a brief so we know what is going on in the complement system. So first, the C1 or the C, it refers to the complement. So we have C1, C2, C3, and until C9, and the order of, or the number here, it does not follow the order by which the molecules are used in the cascade. It follows the order by which these complement proteins were discovered. So C1 is the first protein that was discovered, and C9 is the ninth protein that was discovered. So when we study the cascade or the complement cascade system, things does not go in order. So here, for example, in the classical pathway, it starts with the C1 complex, but then we have the C4, C2 that binds together to, and this complex is responsible for activating the C3. So things are not in order. The other thing we see here, the A and B. So when the complement protein is activated, it usually divides into two fragments. So they decided to call the big fragment as B and the small fragment as A. So when we see C2A, this means that the second complement, and this is the big fragment, the small fragment of the second complement protein. And when we say C4b, this is the big fragment of the fourth complement protein. In the classical pathway, we actually need antigen-antibody complex. So antibodies are already produced and they are bound to the antigens. Now this complex is recognized by the C1 protein. Other C1R, C1S are bound to the C1 to form a C1 complex. This would activate the C4 and the C2. Activations means cutting them into two pieces, big one and small one, the A and B parts. This A and B parts, as we can see here, they form a protein that would cut or activate the C3. So we call the C2A, C4B, we call it C3 convertase because it converts or it cuts the C3 into two fragments. Now, one of the most important fragments that we are going to consider is the C3b. So in the alternative pathway and in the classical pathway, we will meet here. We will meet at the point of the hydrolysis of the C3 or the cutting of the C3 into C3b and C3a. The important molecules that we need to remember is the C3b, because the C3b would activate the C5. The C5, activating C5 means cutting it into two pieces, a small piece and a big piece. We will take the big piece, which is the C5b, that will bind with the cell, and then it will order the other complement proteins C6, 6, 7, 8, 9. So now after the complement 5, things will go in order. So we'll go into C5, 6, 7, 8, 9. And this will form the membrane complex attack here, which I'm going to talk about later. So in the complement system, whether in the classical pathway or the alternative pathway or the mannan-binding lectin pathway, it all indicates the differences on how the cascade is initiated, but all of them will meet at this point. Will meet at the conversion of the C3 into C3b, C3a, having C3b to cleave C5 and to have C5 to initiate forming the membrane attack or the membrane attack complex. Why do we need to go into these extensive steps? Why the C1 complex does not have the ability to just go straight and conduct its function? The reason for that is we need several breaks. We need several control points or checkpoints before we have this end result. If the complement system can be initiated so easily, and if it is once initiated cannot be stopped until the last end step here, then this membrane attack complex can bind to any cell, maybe your body cell. So this would initiate an immune reaction for just any minute trigger. So activating and initiating the complement system cascade need to be tightly monitored and controlled. So we have different breaks or different control checkpoints at each step here. So if this one was bypassed for any reason, it can be caught in this stage. If this was missed, or if this was escaped by any technique or any deficiency in any sort of proteins, it can be caught and corrected at different level until we reach to the final level. So having this long cascade of events, having all of these molecules involved in activating the complement system will provide us with different checkpoints to make sure that the complement system is initiated only when it is actually needed. Returning back to the membrane attack complex, what is the membrane attack complex? Simply, once the C5b bind to the membrane, it will attract other complement proteins, and these complement proteins will just line next and around to the C5b, will form a hole in that cell. The presence of a hole would allow the influx of water inside the cell, and the cell would burst. So this is one of the mechanisms or techniques that complement system follows to conduct its function. So, briefly, these are the three pathways that are used to activate the complement system. The classical, which is the original one that was initially known and discovered. Then they discovered the alternative pathway, and they thought that, okay, that's it, there's an alternative pathway. Finally, and lately or recently, they discovered another way or a third way, which is the mannose binding lectin pathway. In the classical pathway, we need antigen-antibody complex. Here, just we need the pathogen surface antigen, and that's it. And in the lectin binding, or the mannose binding lectin, we need a lectin binding to the pathogen surface. A lectin is a protein, and that can recognize and bind to pathogen surfaces. So in either way, different mechanisms are used to activate the complement system. One of the functions of the complement system is the one that we saw earlier, death of a pathogen agent by forming the membrane complex that will make a hole in the antigen or in the bacteria surface. The other methods, the opsonization and the recruitment of inflammatory cells. So this is this is how opsonization takes place. Remember that we have C3b. We need this C3b to activate C5. So what happens to the C3a, that small particle? It would not be lost. Actually, it would be used in other function. It will bind with the bacteria surface, and it will induce opsonization. It will act as opsonins. And opsonization means putting a flag or marker on the pathogen surface to make it more visible to immune cells. So these, when we activate the complement system, part of that complement system, the B or A part, would be involved in continuing the cascade. The other part is used for opsonization to recruit immune cells or to attract immune cells toward that pathogen. So this is one of the mechanisms that the complement system used to induce its immune function. The other part is these opsonins, or these parts of the complement system, they will act as chemoattractants. So when a white blood cell, for example, a neutrophil, is moving in the bloodstream here, the presence of certain cytokines and the presence of certain concentrations of the complement system, it will act as a chemoattractant to the neutrophils here, because it has receptors for that particular complement protein components. So it will act as a chemoattractant. It will attract these neutrophils to come out of the blood cells and move toward the site of the inflammation. It also will act to activate other white blood cells, for example, mast cells, to start degranulation of the mast cells and producing histamine, creating an inflammatory response. Again, here, for example, clearance of immune complexes, these small, tiny parts of the complement system can bind to antigen-antibody complex. Again, it will act as opsonins. It will flag these antigen-antibody complexes so they are more visible to phagocytes and dendritic cells, and they would be attracted, the phagocytes would be attracted to this labeled and opsonized antigen-antibody complex, and it will be phagocytosed. So these are the three mechanisms the complement system work to enhance or complement the action or the function of the immune system. This is an example of control mechanisms of the immune system. One of them is the C1 esterase inhibitor. So remember the C1 complex that starts the classical pathway. So this is a C1 esterase inhibitor. It acts as a brake, as a control mechanism at the very initial step of the classical pathway. Another example is factor I and factor H. They control the C3b. So C3b is the one responsible for activating the C5, which is the initial complement protein that would form the attack membrane molecule. So factor I and factor H will prevent the haphazard binding of the C3b to the C5. Another example is the protectin and decay accelerating factor, DAF. This molecule is actually a protein, a membrane-bound protein. It binds to the cell membrane of the cells. So presence of the DAF will prevent the accidental binding of the membrane attack complex, MAC, that is formed by the complement system. It will prevent its accidental binding to the cell. Let's see what happens when something goes wrong with the control of the complement system cascade. To appreciate the importance of the control system, let's discuss what happens when the C1 esterase inhibitor is lost. And this occurs when a patient is diagnosed with hereditary angioedema. In this condition, the patient does not have the adequate amount of the C1 esterase inhibitor. Therefore, there is no inhibition, no control in the first step of forming the C1 complex. Therefore, immune reaction can take place for any trigger. So these patients complaining from hereditary angioedema, for no obvious reason, there is no change in their diet, they did not ingest any type of medication, they were not affected with any type of antigen, all of a sudden they develop an allergic reaction. The images here shows the angioedema affecting the lips, but actually it is not restricted to the lip. All parts of the body are involved. The face is the most commonly affected site, including the lips, the eyes, but all types of tissues are affected. The respiratory mucosal the nasopharyngeal area, the laryngeal area would be affected, the respiratory pathways would be affected, and sometimes it might be life-threatening condition, particularly when the patient cannot breathe because of the swelling in the respiratory pathways. The GI mucosa would be affected, and this will lead to abdominal pain, cramps, and different signs and symptoms. So in hereditary angioedema, the T cells work perfect. The B cells are just perfect. The patient had all the immune cells functioning fine. The problem is just with one protein that controls the complement system. This absence of that control or that break, which is the C1 esterase inhibitor, this would make the complement system easily triggered, easily activated, and will lead to these different clinical pictures ranging from just simple angioedema to a severe life-threatening condition affecting the respiratory pathways. Those patients usually are treated with synthetic androgens like danazol, which works by increasing the serum level of the C1 esterase inhibitor, and in cases of emergency where the breathing or the airways are affected, the patient would be given a frozen plasma of C1 esterase inhibitor to control the excessively active complement system. So by that we finished talking about the complement system, and now we're going to talk about the opsonins. We've mentioned the opsonization thing before. So one of the functions of the complement system is opsonization. In opsonins, they do just the opsonization function, which means adding a flag or marking the cells or the areas that are involved in inflammatory reactions, but the opsonins does not conduct other functions of the complement system, including attacking the pathogens, lysing the pathogens by forming a membrane complex, or other functions conducted by the complement system. It does not do that. It just does the opsonization thing. There are different proteins that are used as opsonins. The famous one, the most famous or well-known one, is the C-reactive protein. We know C-reactive protein when we order it. We order it if we are suspecting certain autoimmune or immune abnormality or dysregulation. So we'll ask for C-reactive protein. For example, if a patient is complaining of joint pain and we want to see if he might have rheumatoid arthritis, we will order rheumatoid factor. We will also order C-reactive protein. So C-reactive, what is a C-reactive protein that we are ordering? A C-reactive protein is a protein that is produced from the liver, and the production of the C-reactive protein is enhanced when there is a condition or abnormal condition in the body. This condition does not need to be specific. Any abnormality in the body system will give the order to the liver to produce C-reactive protein. This might include the entrance of a pathogen. This might include presence of tumor cells. This might include presence of atherosclerotic plaque in the blood vessels and in the arteries. Actually, C-reactive protein was found to be a more important marker of cardiovascular diseases, even better than the triglyceride or the cholesterol level in the blood. So this is about this is an example of the opsonins, which is a C-reactive protein. So now we know what is a C-reactive protein when we order it. Last but not least, the cytokines and chemokines. Cytokines and chemokines, again, they are proteins produced by different types of cells, and they aim to steer the immune reaction, either by inhibiting it or increasing and activating the immune response, steering it toward more of a cellular or more of a humoral immune response. So it actually directs the immune response to different directions. These proteins, they have different properties. They share different properties. First of all, one single cytokine can affect different cells and can conduct different or stimulate different actions. So, for example, cytokine 1 can induce proliferation in cell 1, while it enhances differentiation in cell type 2. It regulates the action of cell type 3. So it has different functions. There is no one specific function or no one specific action of a single cytokine. The action, it depends on the different cells that respond to that cytokine. The second property is the synergism, which means that when cytokine 1 produces level like extra one step in differentiation, and cytokine 2 will also increase one step in cell differentiation, having the two cytokines might increase it to 4 or 5 steps in differentiation. It's not just the addition of the effects of the two, which is very handy when we need a powerful immune response. The last property is that the cell or the cytokine might have an autocrine level or autocrine function. And in autocrine, the cytokine will bind to the cytokine receptor that is present on the same producing cell, and this would enhance the function of the cell and will make sure that the cell continues its function for a longer duration. The paracrine level, where the cytokine produced by one cell would affect the other cell, like, for example, enhancing stimulation, enhancing proliferation, or differentiation. And the third effect is the endocrine, where the cytokines produced will go through the bloodstream and affect distant organs to conduct certain functions or produce certain enzymes. So we are not going to talk more about the cytokines, but we are going to mention them whenever we go discussing whether the cellular part of the humoral response or the adaptive immune response. You will see how the cytokines produced will steer the function and the action of the immune system. Cytokines include interleukins, interleukin 1, 2, 6, TNF, tumor necrosis factor, for example, epidermal growth factor. All of these are cytokines responsible for differentiation, responsible for stimulation, maybe inhibition, maybe suppression. So it has different functions that we are going to mention in the next lectures. So by the end of this lecture, we have been introduced to different components to the humoral immunity. The interferons, now we know them, the microcidal molecules, complement system, opsonins, cytokines, and chemokines. In the next lecture, we will start talking about the next component of the innate immunity, which is the cellular innate immune response. This is the end of this part, and we'll see you next part talking about the cellular innate immune response. Thank you.